Hello and welcome to video 4, Cell Life Cycles. This is Mr. Troy, and today we're going to be exploring the wondrous life of cells and really how they end up growing and growing and, spoiler alert, at the end of their lives, divide and make anew. So yes, cells do have lives, kind of like teachers, except I guess I'm not one to talk since I just finished like a 1,000-page book on music theory and math and how they're related, and now I think I understand less of how the world works than I did before. Music? Math? Like what? Anyways, we're moving on. Okay, so I do want to talk a little bit about unicellular, which is like single-celled organisms, versus multicellular organisms, cellular. And so cell division for unicellular, single-celled organisms, like prokaryotes and some eukaryotes, like amoebas, these are going to be using cell division mainly just to divide themselves. So we have like one becoming two new cells. It's essentially a cloning process. So bacteria do this all the time, and again, some eukaryotes like amoebas will do this too. On the other hand, multicellular organisms like you, me, plants, fungi, anything that's made up of more than one cell, these are going to be using cell division for three main things. First off, development, right? So once we're like, so this is like a sand dollar embryo, so like this is kind of, imagine a cell, like a sperm cell went into that egg cell. That one cell needs to eventually divide to make multiple cells to have something to work with. So developing the embryonic stage requires mitosis or cell division. Growth, right? You are obviously very different than you were about 15 years ago, and so it's important that mitosis helps us divide new cells to have us actually physically get bigger and grow. And of course, repair, super important. Like when you're playing dangerous sports like handball, and you know that ball grazes your side and now you have this big gash on your side and you're like, empty space of wound, and you know you need mitosis or cell division to fill in that empty space with new cells. Hence why scars have slightly different color tones, because those are actually new cells being made for you to fill that gap. So mitosis or cell division is extremely important for both unicellular and multicellular organisms, except we get to use them for more stuff because, I mean, in a way we are slightly more complicated. That being said, this is the main picture I want to focus on today. This is a cell life cycle. Super cool. I will teach you how to read it in like 2 minutes now. But before we do that, we can divide the cell life cycle into two stages. We can divide it into interphase. That's a T, interphase. Interphase. And we can divide the other part into the mitotic phase. Mitotic phase. Okay, so the mitotic phase and interphase are going to be the two main phases of the cell life cycle, but each of those phases have smaller phases in them. So the interphase includes G1 or gap 1. It also includes S phase, and it includes G2. Okay, and this is going to be happening about 90% of the time for the cell's life. Okay, so interphase is like the big part of life for cells. And then I cannot talk today. The mitotic phase is going to be basically mitosis, which is the actual dividing up of the cells, and cytokinesis, which is the physical splitting of two cells. And so a lot of textbooks will kind of pair mitosis and cytokinesis into one sort of phase called the mitotic phase, and that's what we're going to be covering. That being said, I do want to go into a little bit of detail of how the cell life cycle works. Not too much though, because you will be covering that through a project. Hey! So we're going to start here with the G1 phase. So this is G1 or gap phase. A lot of people confuse G for growth instead of gap, because that's what the cell's doing in the G1. It's growing. And I should add physically. Okay, so it's physically growing in this stage, because you have to imagine it just came out of the M phase, right? Just came out of the M phase or mitosis. And so we actually have a pretty small cell at this stage, because we've just gone from one cell to two cells. So this new small cell needs to actually physically grow and make its proteins and make sure that it has all its organelles before it enters the rest of its life. And so again, in G1, you're physically growing. You can think of it as a growth phase, but really it's called gap 1 phase. Now there is a auxiliary step right here called the G0 phase, where cells actually stop dividing. They kind of leave this cell life cycle. Okay, and so these cells are going to be like your brain cells, the neurons, that don't really divide. We do now know that there are some neurons that divide, but for the most part, most of your neurons go into this G0 phase and just kind of stay the same. They don't keep dividing themselves. So some of your neurons are your oldest friends. Haha. Okay. Alright, moving on. We then go into the S phase, right? And so S actually stands for synthesis. And the reason why is this is where DNA replication is happening. So right here I'm just going to write DNA replication. And so in this stage, we're actually creating an entire new copy of your genome, or all your DNA. So in this phase, like you have 46 chromosomes, right? So if I drew 46 of these, each of them is going to have a duplicate piece synthesized through DNA replication, which again we will cover together. And so in the end, you double the amount of DNA. You don't actually double the number of chromosomes, we'll talk about why that happens, but you do double the amount of DNA you have. Next, in the G2 phase, which is also called gap phase, so you have the G1, G2 phase. In the G2 phase, we're just getting ready. So this is the prep before division stage. And there are some important factors in G2 that we can go over, but really, all I really want you to know is that in G2, there's just a major checkpoint right here that's going to happen before we can enter cell division. Cool? Okay. So, those are all the stages of the cell life cycle. Finally, in mitosis, which we'll cover in a different lesson, that's where the cells physically divide and become anew. Hey, so I want to focus a little bit more on the control system of the cell because we are interested in cancer for this project. It turns out that there are multiple checkpoints within mitosis, some of them being the G1 checkpoint. So the G1 checkpoint. So you kind of need to go through, oh, hey, stop. You kind of need to go through this checkpoint to pass G1, so this is all G1, right? There's another checkpoint right here in G2, which is right before you go into mitosis or cell division. Very important for making sure you don't get cancer, right? Because you don't want uncontrolled cell division right here. And then finally, there's also a mitosis checkpoint before we can continue on into G1 phase and finish off mitosis. And so this is really cool because these checkpoints kind of stop our cells from dividing uncontrollably and eventually making cancer. But today we're going to focus on one checkpoint in particular, and that's going to be the G2 checkpoint right here. So this is the one we're going to focus on. So in your notes or something, I would just write G2 checkpoint, important, or something. Okay. So the G2 checkpoint is going to rely on three main players: lion, tigers, and bears. Just kidding. So G2 checkpoint. Checkpoint. Okay, so the three main players that I want to focus on today are cyclin, CDK, and MPF. What are these? Well, I will tell you. Cyclin is going to be a protein, actually. So this is my cyclin, it's a protein. And cyclin is a protein that will fluctuate in concentration inside the cell, depending on what stage of the cell cycle your cell is in. CDK, on the other hand, is an enzyme, which is also a protein, in this case. So this is an enzyme. And you might be able to see it now, but CDK is an enzyme that will actually bind with this cyclin to create our MPF, which is basically the complex we refer to when cyclin and cyclin-dependent kinase is put together. So this is my cyclin, and this is my CDK, and combined together they make MPF. And what the heck does MPF stand for? Well, let me tell you. It stands for maturation promotion factor. Okay, and so you need this MPF, to pass G2 checkpoint. Okay, so one more time, cyclin is a protein that's going to kind of change its concentration throughout the lifetime of the cell. Cyclin-dependent kinase is an enzyme that kind of stays the same concentration throughout the life cycle of the cell. And when they combine together like a Megazord in Power Rangers, you get this cyclin-dependent kinase CDK combining with cyclin to make MPF, and MPF is kind of the all-pass to get through G2. So what that looks like here is basically, I'm going to try to refer this back to the checkpoint we were talking about before. Actually, you know what? Let me just go back to that picture. Hey, why not, right? Oh, too far. So right here, it turns out that the cyclin concentration, right, so the protein concentration of cyclin, that's going to be very low, right, very, very low through most of G1 and a little bit of S phase. But cyclin concentration starts increasing, so start making more cyclin. Starting in S phase, so about middle of S phase, we start making a bunch of cyclin protein. And eventually, this cyclin protein is going to keep getting more and more, right? The cyclin proteins can get more and more until we finally have enough to bind with the CDKs that are just kind of normally hanging out in the cell. So the concentration of CDK doesn't change, it's staying the same in the cell. Okay? And it's only during S phase that we get more and more cyclin to bind with the CDK and eventually have enough to make our MPF right here so we can pass through into mitosis phase. So I'm going to show you guys a slide, and I would just pause on this slide right here. You can see how cyclin, right, the concentration of cyclin kind of starts in S phase to go up, all right, and then as soon as it reaches its peak, we have maturation promotion factor activity, allowing us to pass into M phase. It should be noted that cyclin is degraded during M phase. It's actually anaphase in mitosis, but we can go over that later. And so basically, we don't have more cyclin start being made until S phase, and as we make more and more and more, they bind to the CDKs already in the cell. If you would like to, you can just kind of pause this part in the video and just like look at it and be like, hey, that's cool. And read through all these descriptions. But essentially, I do want you to know how cyclin and CDK play a role in passing you through G2, okay, the G2 checkpoint. And it's important because the way it's kind of done is that you need to go through S phase to start making cyclin, which means that the cell, to pass through G2, must have made its DNA. There are some other ways that cell division is regulated. For instance, we do know from laboratory experiments that crowded cells stop dividing. So if you actually have too many cells in one area, they'll usually stop dividing by sending out some growth factors to each other, growth inhibiting factors. So this is called density-dependent inhibition. Also, it turns out that cells need to be attached to a surface that has nutrients. It's called a substratum, and that's going to be called anchorage dependent. So if you see right here, the cells actually stop growing once they reach a single layer, and if you remove some cells, they'll fill in that space. Right? So this is density-dependent inhibition, where if you have too many, they'll just stop growing. But if they have some space, they'll grow. Cancer cells don't exhibit either of these inhibitions or dependences, and so if you notice right here, cancer just kind of goes like, hey, I'm just going to build on top of you, like, and go out of control. So they, again, do not exhibit anchorage dependence or density-dependent inhibition. So basically, when you cannot control the cell division cycle, life cycle, that's when you get cancer in cells. And tumor formation is when you have a bunch of cell growth at once without stopping. This malignant tumor idea is that you can actually have metastasis, so this is better shown through this right here. So if you notice right here, this is a tumor growing, right, from a single cancer cell, so you've got uncontrollable cell division there. This will eventually grow bigger and invade neighboring tissue, and once this actually spreads to the lymph and blood vessels to other parts of your body, that's when you call it metastasis, because these new cancer cells can grow in other parts and establish new tumors, and eventually that's around stage 3, stage 4, and where treatment gets a little dire. And so, if you were confused about any of this or are interested in learning more, I would check out the Crash Course Biology series or Bozeman Biology for more references. With that said, this is under 15 minutes. Thank you very much. Have a great day.